Welcome to the deep dive.
Yeah.
Imagine this, a virus, one that causes serious neurological problems. It kind of fades away for decades, and then boom, it suddenly reappears.
Yeah, that's basically the story of Western equine encephalitis virus, WEBV. It's an arbovirus transmitted by mosquitoes.
Right, and people mostly thought it was, well, a thing of the past.
Pretty much. But then in 2023, totally unexpectedly, it reemerged down in South America. We saw over 200 human cases and something like 2,500 horse cases.
Wow, that's quite a comeback and a really troubling one.
It really is. I mean, this virus was thought to be getting less virulent, certainly less of a human threat recently. So this resurgence, it's a major public health flag, shows how fast things can change with viruses.
Absolutely. So today, our mission is to dive into the hidden molecular details. How does WEEV actually infect different hosts? And maybe get some clues about these shifting disease patterns.
Exactly. We're going to look at how really tiny genetic tweaks in the virus's surface proteins basically decide which cells it can get into.
And why figuring that out is so vital for, well, being ready for the next outbreak.
And we're using some cutting-edge stuff here. Detailed images from cryo-electron microscopy, lots of mutational analysis. The goal is to give you a really clear picture of this molecular dance.
Connecting those tiny details to the big public health picture.
That's the plan.
Okay, so let's start with the basics. What exactly is WEEV? Why the big concern?
Well, it's an alpha virus. That family, it's kind of notorious for causing encephalitis, brain inflammation in both people and horses.
And it's nasty stuff, right?
Yeah.
It can leave lasting damage.
Yeah, absolutely. Survivors often face long-term neurological problems. And historically, WEEV was a major worry, especially back in the early 20th century with big outbreaks in North America.
And then, like we said, it just sort of faded. Why did that happen?
That's the question, isn't it? Cases just steadily dropped off. The last documented human case in North America was way back in 1999. It really seemed like it had gone underground, at least for humans.
Which makes that 2023 South American comeback so jarring, like a ghost reappearing.
Exactly. So how does it actually cause disease? It has to get inside our cells.
Right. How does it manage that? What's its molecular door key?
Good way to put it. Like a lot of viruses, it has proteins on its surface that are designed to latch onto specific receptors on our cells. Think of it like a key fitting a lock.
Okay.
For WEEV, the keys are these glycoproteins. They're called E2 and E1. They work together on the virus's outer shell.
And the main lock on human cells, what's it targeting?
The primary one seems to be a molecule called protocaterin 10, or PCDH10 for short.
PCDH10. And what does that do normally?
Well, what's really interesting is that PCDH10 is an adhesion molecule. It helps cells stick together, and it's found a lot in the brain. It's actually important for how brain synapses develop.
Ah, okay, so that immediately clicks, right? If it's targeting something crucial in the brain, that explains the neurological illness.
Precisely.
And you mentioned this receptor, PCDH10, it's kind of unique.
It really is, yeah. Among the alpha viruses we know about, PCDH10 is unique to WEEV. It doesn't look anything like the receptors used by its relatives, like Eastern equine encephalitis, EEEV, or Venezuelan equine encephalitis, VEEV.
So WEEV found its own special door to unlock. It's not using the family key, so to speak.
That's a good analogy. It has its own specific target.
So how does that interaction work? The E2, E1 key fitting into the PCDH10 lock. What does that look like at the molecular level?
Okay, so the virus's E2-E1 complex binds very specifically to one part of PCDH10, a region called the EC1 domain.
EC1.
Right. And the contact is extensive. It's like two perfectly matched puzzle pieces clicking together. They bury a pretty large surface area when they connect, about 1,500 square angstroms.
Right.
That means it's a strong, stable connection.
Okay, a really tight grip. And this led to a kind of aha moment in the research, didn't it? Something about PCDH10's normal behavior.
It really did. Quite fascinating. Researchers realized the way WEEV-E2 binds to that PCDH10 EC1 domain is incredibly similar to how PCDH10 normally interacts with itself.
With itself? How so?
PCDH10 molecules usually pair up, forming what are called anti-parallel homodimers. That's just part of their normal job in the cell, helping with cell adhesion.
So the virus is basically mimicking a natural interaction, hijacking a binding site that PCDH10 uses to connect with another PCDH10. That's clever.
Extremely clever. And what this strongly suggests is that one WEEV can probably only bind to single unpaired PCDH10 molecules, monomers.
The pairs.
Right. Because when PCDH10 is in its paired or dimeric form, that binding site the virus needs is likely blocked. occupied by the other PCDH10 molecule.
Ah, I see. So the virus needs to find PCDH10 when it's sort of available and not already paired up.
Exactly. It needs that specific monomeric form to get in. It's a really fundamental insight into how it targets the nervous system with such precision.
Okay, that's a neat viral trick. But it also brings up the big question. If it's so specialized for this receptor, how did its infection patterns change so much? And the decline, the reemergence?
Yeah, that's where the virus's own evolution comes in. The answer seems to lie in the different genetic lineages of WEV that have developed over time.
Lineages. Okay, tell us about those.
So, broadly, we classify WEV strains into lineage A and lineage B in North America. And lineage B has subgroups, B1, B2, B3.
Right.
And then there's the newly recognized lineage C, which is what popped up in South America recently.
And do these different lineages use receptors differently? Does that explain their different impacts?
It seems to, yes. So the older, really virulent lineage A strains, like the Macmillan strain from the 1930s, 40s, they were quite versatile.
How so?
They could bind to both human PCDH10 and another set of receptors, the LDLR-related proteins, specifically VLDLR and ApoER2.
Okay, so they had multiple keys, essentially.
Kinda, yeah. But then if you look at more recent lineage B3 strains, like one called Imperial 181, isolated in 2005,
Yeah.
it seems to have lost the ability to bind to the mammalian versions of PCDH10, VLDLR, and ApoER2.
Lost it completely.
Yeah.
So Imperial 181 basically couldn't infect humans or horses effectively anymore.
That seems to be the case for mammals. Interestingly, though, it could still bind to the avian version of PCDH10, like from house sparrows, and also garter snake receptors.
Huh.
And in lab animals, Imperial 181 is non-pathogenic. It doesn't cause disease.
So this shift, losing the ability to bind human or horse receptors, that could explain the big decline in North American cases.
It's a very strong explanation, yes. The virus essentially adapted away from being a major mammalian pathogen in those lineages.
Now here's the really fascinating part for me. What caused such a dramatic change in binding? Was it a whole bunch of mutations or something smaller?
This is where it gets really cool. The research pinpoints it down to potentially a single amino acid substitution in that E2 glycoprotein.
One single change.
Yeah, at position 149 in the E2 protein. Virulent strains like Macmillan typically have an amino acid called leucine, or L, at position 149, L149.
Okay.
But Imperial 181 has glutamine, or Q, at that position, Q149.
And that one change, L to Q.
That L149Q mutation seems to be enough to disrupt the key contacts needed to bind tightly to human PCDH10.
Wow. So one tiny typo in the virus's genetic code basically slammed the door shut on human infection for that lineage. That could explain decades of near silence in North America.
It's a very compelling molecular explanation for a large-scale epidemiological pattern.
But wait, you said Imperial 181 could still infect sparrows. How does that work if its human key was broken by that Q149 change?
Good question. It turns out the sparrow version of PCDH10 is slightly different. It has what we call compensatory contacts. Meaning, even though the virus changed at position 149, the sparrow receptor has different amino acids in its binding site, like at positions R89 and L74, that can still make favorable connections with the virus's Q149.
So the sparrow lock could still accommodate the slightly changed key.
Exactly. It highlights how these interactions are a two-way street, and sometimes changes in the host receptor can compensate for viral mutations. You need enough favorable interactions for binding to occur.
That's amazing adaptability. And this discovery about the E2L149Q polymorphism, it has real practical implications for tracking the virus, doesn't it?
It absolutely does, because now, just by sequencing the E2 gene of a WEV sample,
You can predict.
you can make a pretty good prediction about whether that specific strain is likely compatible with human PCDH10 receptors. It gives us a sequence-based way to assess potential risk.
That's huge for surveillance.
Yeah, right.
Okay, so besides PCDH10, you mentioned those other receptors, VLDLR and ApoER2. Does WEV use the same strategy for binding those, or is there variation there too?
Oh, there's definitely variation there too. It's not one size fits all. Even among the virulent strains that can bind these LDLR-related receptors, they've evolved different ways to do it.
Different strategies for the same goal.
Exactly. Take that ancestral Macmillan strain again. It needs to engage the VLDLR receptor at two specific sites simultaneously using parts of both its E1 protein, K227, and its E2 protein, K190, for really efficient binding.
Okay, a coordinated two-point attachment.
Right. But then there are other lineage A strains, like one called Fleming. It binds VLDLR using a completely different site on its E2 protein, E2K81. This site is near what's called the virus's threefold axis, and in other related strains, that position is occupied by a different amino acid, glutamate, which wouldn't work for binding.
So different strains evolve distinct docking mechanisms for the same type of receptor. It's like having different keys that somehow still manage to open the same lock, but using different parts of the keyway.
That's a great way to think about it. It really shows the diversity of evolutionary solutions viruses can come up with to solve the problem of getting into a cell.
Okay, this is deep molecular detail. Let's bring it back up. How does all this connect directly to pathogenicity, the virus's ability to cause disease, and what we need to do about it? What's the payoff for knowing this?
Well, the direct link is location, location, location. Both PCDH10 and those LDLR-related proteins, VLDLR and ApoE-R2, are expressed on brain cells, on neurons.
Right.
So being able to bind to them is fundamental for the virus to actually infect the brain and cause encephalitis. Imperial 181, remember the one that lost mammalian receptor binding?
Yeah.
It replicates really poorly in mouse brains in experiments, and it's non-pathogenic, so there's a very strong correlation. No binding to brain receptors, no brain disease.
And scientists actually tested this connection directly.
They did. It's quite elegant, actually. They took the non-pathogenic Imperial 181 virus and genetically engineered it. They put back specific E2 mutations.
Like changing the Q149 back to L149.
Exactly, to restore PCDH10 binding. Or they made other specific changes, like adding the K81 or K190 mutations to restore VLDL-RAPOER2 binding, mimicking the Fleming or Macmillan strains.
And what happened?
Remarkably, putting those specific keys back restored the virus's ability to infect neurons in lab models.
Wow. Direct proof.
Yes, it confirms that these specific receptor binding modes are directly responsible for letting the virus into neurons. And interestingly, it also showed that different strains might rely more heavily on one receptor than another. A Macmillan-like mutant, for example, seemed primarily dependent on PCDH10 for getting into neurons, while a Fleming-like mutant seemed to rely more heavily on the LDLR family receptors.
So knowing the E2 sequence lets us predict not just if it can infect neurons, but maybe how it prefers to do it.
That's the implication, yes. It gives us a much more nuanced understanding.
So how does this predictive power help us understand the recent South American outbreak? What do the sequences of those lineage C strains tell us?
Okay, so looking at those recent strains like EQ1090 or DLAV218 from the 2023-2024 outbreak,
Yeah,
their E2 glycoprotein has L149.
L149, the one that binds human PCDHN.
Exactly. That tells us immediately they are capable of binding the key human receptor, which fits perfectly with that they're causing human cases.
Okay, makes sense. What about the VLDLR binding?
Interestingly, these South American strains lack the key amino acids like K81 or K1968227 that are needed for efficient VLDLR or ApoER2 binding.
So they have the PCDH10 key, but not the VLDL-VLRAPOER2 key.
That seems to be the case, and lab experiments confirm this prediction. They bind PCDH10, but not VLDL-RAPOER2. This helps explain why they seem to be persisting in animal cycles, epizootic circulation, while retaining that ability to jump into humans via PCDH10.
It's like they kept one crucial key, but maybe shed another. Fascinating. What about back in North America? Is evolution still happening there?
Oh, absolutely. It never stops. Some of the more recent North American lineage B strains, like BFS 09997 and EP6, have actually picked up mutations, specifically E2K181, which seems to restore their ability to bind VLDL-RAPOER2.
Even though they might have other mutations that would normally hinder it.
Exactly. It suggests compensatory evolution is happening. The virus is finding ways around potential roadblocks, regaining abilities it might have lost. It's a constant evolutionary arms race.
And this detailed receptor knowledge isn't just for WEEV itself. It sheds light on related viruses, too.
It does. There's another related alpha virus in North America called Highlands J virus, HJV. Research shows it also uses PCDH10 and another receptor called avian MXRA8, but apparently not VLDL-RAPOER2.
So some similarities, some differences.
Right. And even in HJV, just a single amino acid change in its E2 protein, A177K, was shown to restore binding to horse PCDH10. It just underscores how incredibly precise these molecular interactions are and how small changes can have big effects across the viral family.
Okay, bringing this all together, we understand the keys, the locks, the evolutionary shifts. What does this mean for actually fighting these viruses? Is there a therapeutic angle here?
Yes, definitely. If we understand that binding to PCDH10, VLDLR and ApoE2 is critical for WEEV causing disease, especially brain disease.
Then maybe we can block that binding.
Exactly. And this research points to a really promising approach using a decoy receptor.
A decoy, how does that work?
Researchers engineered a piece of the VLDLR receptor, specifically the LA domains 1 and 2 that the virus binds to, and fused it to part of an antibody, creating something called an FC fusion protein.
Okay, so it looks like the real receptor to the virus.
Precisely. It acts as a false target. The idea is the virus latches onto this decoy protein floating in the bloodstream instead of latching onto the actual receptors on brain cells.
A molecular trap. Does it work?
The preclinical results are actually quite impressive. In mouse studies, giving a single dose of this VLDLR LA1-2 decoy before infecting them with a lethal dose of the Macmillan WEEV strain, it protected 80% of the mice from dying. In the control group that didn't get the decoy, survival was 0.
Wow, 80% protection from a lethal dose. That's significant.
It is. And what's more, this decoy seems to have broad activity. It works against the virulent WEEV strains that use VLDLR, but it also showed activity against EEV, that other nasty alpha virus.
So it could potentially be useful even if WEEV evolves further.
That's the hope. It suggests that even if new WEEV variants pop up, perhaps ones that regain that VLDLR binding ability.
Like those compensatory mutants you mentioned?
Right. This decoy strategy could still be a viable and effective therapeutic option. It's a proactive approach, grounded in this deep molecular understanding, offering a potential tool to fight back.
So wrapping up, hopefully this deep dive has given you a much clearer sense of how these really subtle changes down at the molecular level in the virus's E2 protein can have these huge consequences.
Yeah, affecting which hosts it can recognize, how it spreads, whether it causes disease. It's quite a story.
From WEEV fading in North America because it lost the ability to bind mammalian receptors to its sudden reemergence in South America because those strains kept the human PCDH10 key. It's viral evolution playing out right in front of us.
And these insights, they really do give us a way to sort of gauge the potential threat from different WEEV strains just by looking at their genetic sequence, particularly E2 and E1. That's incredibly valuable for surveillance and preparedness.
So the final thought for you, our listener. What does all this mean going forward? How might understanding these tiny molecular shifts in a virus like WEEV change how we approach global health surveillance?
Especially for other emerging viruses, arboviruses, where maybe similar evolutionary games are being played kind of under our radar right now.
How does this knowledge shift how we think about that delicate balance between a virus and its hosts, and maybe our ability to anticipate what might emerge next?